Plants require light for photosynthesis, a process in which light energy is utilized to synthesize carbohydrates from carbon dioxide and water. The major pigment that absorbs light for photosynthesis is chlorophyll. Besides photosynthesis, light also regulates several growth and developmental processes in plants. If a dicot seed is germinated and grown in complete darkness, the hypocotyl will grow longer, cotyledons will not expand, and there will be no formation of chlorophyll. If the same seed is grown in presence of light, the hypocotyl will be shorter, cotyledons will expand and become green by forming chlorophyll. These changes brought about by light, which are independent of photosynthesis, are called photomorphogenesis. The major photoreceptor for photomorphogenesis is phytochrome. Phytochrome absorbs red and infrared light. The red absorbing form is referred to as PR form and the infrared absorbing form is the PFR form. These two forms are photoreversible. PR form changes to PFR form by absorbing red light and the PFR changes to PR by absorbing infrared. Out of the two, PR is the inactive form and the PFR is the active form. The seedlings grown in darkness will have phytochrome in the PR form. The phytochrome changes to its active PFR form when the seedlings are exposed to light. Since phytochrome is photoreversible and only the PFR form is the active form, photomorphogenic changes induced by red light is reversible with infrared light. Phytochrome regulates several processes in the plant, starting from germination, seedling growth, vegetative growth, reproduction and senescence. Photoreversibility of a red light induced response by infrared light is the primary criteria used for relating any photoresponse to phytochrome. Besides inducing morphologically visible responses like germination, inhibition of hypocotyl elongation, chlorophyll synthesis, etc., phytochrome also induces many biochemical and physiological changes in plants which are not visible. For example, activity of certain enzymes are induced or promoted by phytochrome, and to observe this response, biochemical methods need to be used. Let us see in detail about the structure and function of phytochrome in plants. The first observation of the effect of red light on plants was on photoperiodism, that is, the duration of day length required for flowering in plants. On the basis of photoperiodism, plants are broadly grouped into three categories: the short day plants, which cannot tolerate light more than the critical period of day length; the long day plants, which cannot tolerate less than the critical period of day length; and the day neutral plants, which can flower in any photoperiod. Garner and Allard were the first to observe inhibition of flowering in the short day plant Xanthium when the dark period was interrupted in the middle by a flash of red light. Later, extensive experiments on the germination of lettuce seeds, which required light for germination, led to the discovery of phytochrome. Flint demonstrated that germination of lettuce seed was stimulated by red light and inhibited by infrared light. Bothwick and Hendricks demonstrated that treating the lettuce seeds alternatively with red and infrared light, the seed germination was either induced or inhibited depending on the terminal exposure. Repeated exposure like red plus infrared plus red resulted in germination, whereas an exposure like red plus infrared resulted in the inhibition of germination. In other words, the germination that could be induced by red light was always reversible with infrared light, and the response dependent on the terminal exposure. Two interpretations were possible to explain this reversibility with red and infrared light. One was the possibility that two separate photoreceptors existed in plants which absorbed red and infrared light and acted antagonistically, that is, one that absorbs red light promoting germination and the other that absorbs infrared light inhibiting the same. The second possibility was that there might be a single photoreceptor that can exist in two interconvertible forms, one absorbing red light and the other absorbing infrared light. The work of Butler et al. Provided evidence for the existence of a single photoreceptor named Phytochrome with two interconvertible forms PR and PFR. Butler et al. Demonstrated this unique property of photoreversibility in vitro by isolating phytochrome from plant extracts. Phytochrome was first isolated and purified from dark grown etiolated seedlings of oat. Phytochrome is a soluble protein with a molecular weight of about 250 kilodalton. It occurs as a dimer made up of two subunits. Each subunit of about 125 kilodalton has two components, the light absorbing chromophore part and the polypeptide protein part. The chromophore part is made up of a linear tetrapyrrole ring structure which is attached to the protein through a thioester linkage to a cysteine residue. Chlorophyll also has tetrapyrrole rings in a circular structure with magnesium atom in the center instead of a linear structure like in phytochrome. In the chlorophyll, the circular chromophore part is attached to a phytol chain, unlike phytochrome in which the chromophore is attached to a protein. Because of the similarity in their chromophore structure, both chlorophyll and phytochrome have their peak of absorption in red light. Phytochrome is synthesized in the PR form in the dark grown etiolated seedlings. When the seedlings are exposed to red light, the PR form gets converted to PFR form. The PFR form is converted back to PR form when it absorbs infrared light. The conversion reconversion happens in the PR and PFR forms of phytochrome because of a cis-trans isomerization of the carbon bond between 15 and 16 carbon of the chromophore. PR is the cis form and PFR is the trans form of the phytochrome. The cis-trans isomerization also causes certain conformational changes in the protein part of the phytochrome. Since phytochrome responses are induced by red light, the PFR form is considered as the active form of the phytochrome. The PR form of phytochrome has a major peak of absorption in the red and the PFR form has a major peak of absorption in the far red region of the light spectrum. Both PR and PFR forms also have a minor peak of absorption in the blue part of the spectrum. The absorption spectra of PR and PFR forms show an overlap in the red far red region of the spectrum. Because of this overlap, it is not possible to convert all the molecules of PR to PFR or all the PFR to PR. By exposure to monochromatic red light, about 85% of the phytochrome is converted to PFR. Here, a small part of the PFR is getting converted back to PR by the spectral overlap. Similarly, by exposure to monochromatic far red light, about 97% of PFR is converted to PR. This equilibrium of PR and PFR is termed as the photostationary state of the phytochrome. The photostationary state is the ratio of PR to PFR and can be determined for any type of radiation like blue, yellow or white light. The amount of PFR formed will vary with the type of light used. In nature, the sunlight has more component of red light during sunrise in the morning and sunset in the evening. Depending on the composition of the light received, the photostationary state of the phytochrome will also vary during daylight. Some of the plants use the changes in the photostationary state of the phytochrome to measure the onset and end of the natural daylight. Phytochrome has two parts in its structure, the chromophore part and the protein part. Different types of phytochromes differ in their protein part, although all of them have the same chromophore that absorbs red and far red light. Molecular biology studies have shown the existence of a multi-gene family that encodes phytochrome genes, which have been identified and named as phytochrome A, phytochrome B, phytochrome C, phytochrome D and phytochrome E. On the basis of the products of this phytochrome gene family, two types of phytochromes are recognized as type 1 and type 2. Type 1 phytochrome is coded only by phi A, which is most abundant in the dark grown seedlings. The PFR A encoded by phi A is unstable. In addition to this, when the seedlings are exposed to light, the expression of the gene phi A is strongly inhibited. The remaining genes of the family phi B to phi E encode type 2 phytochromes. They are more dominant in green plants grown in presence of light. The PFR of phi B to E is more stable in light. The phytochromes differing in their protein structure also regulate different functions in the plants. Phytochrome responses can be classified to three types on the basis of the amount of light required to induce a response. These are very low fluence responses, low fluence responses and high irradiance responses. Fluence refers to the amount of light received by the plant and is defined as the number of photons impinging or falling on a unit surface area. Fluence is measured as moles of quanta per square meter. Fluence rate or irradiance is measured as moles of quanta per square meter per second. Very low fluence responses is induced by very low fluence of light between 10⁻⁸ to 10⁻⁴ micromole per meter square. This is very low amount of light and converts only about 0.02% of the total phytochrome to active PFR form. These responses are not reversible with far-red light because far-red light itself produces about 3% PFR due to overlap in the absorption spectra of PR and PFR. Germination of the seeds of Arabidopsis and inhibition of the growth of the mesocotyle, that is the axis between coleoptile and root in a monocot seedling in oat seedlings, are some of the examples of VLFR. Low fluence responses is observed between 1 micromole per meter square to 10 to the power of 2 micromole per meter square fluence of light. Most of the red for red reversible responses like lettuce seed germination, inhibition of internodal elongation in dark grown pea seedlings, and inhibition of flowering in the short day plant Xanthium are low fluence responses. As we have seen earlier, the phytochrome was discovered through these photoreversible responses. Both VLFR and LFR responses follow the law of reciprocity. This law was given by Bunsen and Roscoe in 1850. This law defines the relationship between fluence rate and the irradiation time. Depending on the intensity of light and the time of exposure, both VLFR and LFR responses reach a saturation point after a certain time of exposure. The saturation point is a product of time multiplied by intensity. In other words, the saturation point is reached within a short period of time if a high intensity light is used, whereas a low intensity of light will require a longer time to reach the saturation point. Once the saturation point is reached, further increase in either intensity or time will produce no quantitative change in the response. The high irradiation response differs from both VLFR and LFR in that they do not follow the law of reciprocity. Varying the intensity of light or the time period of exposure changes the response quantitatively. HIR requires continuous exposure to light and the response is proportional to irradiance or brightness or intensity of the light. They are also not reversible with far-red because the response can be induced even under continuous far-red. High irradiance can induce some of the responses which are inducible with low fluence also and are reversible with far-red. Inhibition of hypocotyle in mustard, lettuce and petunia, enlargement of cotyledons in mustard and anthocyanin synthesis in many seedlings are some of the high irradiation response inducible responses. Phytochrome responses are also classified as slow and fast responses depending on the time taken by the plant to respond to a brief exposure to red light. The classical examples of photoresponses to phytochrome like germination of lettuce seeds or synthesis of anthocyanin in mustard seedlings or inhibition of hypocotyl in dicot seedlings are considered as slow responses. Although these responses are inducible with a brief exposure to red light of 1 to 5 minutes, there is an incubation time required before the response can be observed. In case of lettuce seed germination, the seeds are to be incubated in darkness for 24 hours after 1 minute of exposure to red light. Similarly, the dark grown seedlings of mustard are to be incubated in darkness for 24 hours after 5 minutes exposure to red light to allow maximum synthesis of anthocyanin. In contrast to slow responses, the fast responses to phytochrome are those which can be observed within minutes or seconds after a brief exposure to red light. The plant touch me not or Mimosa pudica closes its leaves within seconds when it is touched. This response is due to changes in the turgor pressure of the cells located on opposite sides of the pulvinus, which is a specialized structure at the base of the petiole. The turgor changes depend upon the movement of potassium and chlorine ions across the membrane. Opening and closing of leaves in the same plant can also be regulated by exposing the leaves to red and infrared light. Exposure to red light causes closing of the leaves, and infrared light promotes opening of the leaves. This response, which comes within 5 minutes of exposure to light, is a fast response. Phytochrome regulates the response by causing changes in the movement of potassium and chlorine ions. There are some more examples of fast responses to phytochrome. Red light exposure causes changes in the surface potential of wood coleoptiles. Similarly, in the isolated root tips of barley suspended in a buffer, red light induces efflux of hydrogen ions, imposing a positive charge on the root tips, making them adhere to the negatively charged glass surface. This effect of red light is reversible with infrared light. In accordance with the slow and fast responses to red and infrared light, two separate models have been proposed to explain the mechanism of action of phytochrome. The fast responses induced by the phytochrome are due to rapid changes in the turgor pressure of the cells involving ion fluxes. The rapid changes in the flux of ions across the membrane can also cause changes in the bioelectric potential of the cells. The slow responses like red light induced anthocyanin synthesis or inhibition of hypocotyl growth involve alterations in the expression of genes. These alterations can cause either a positive effect of induction or promotion of a metabolic activity or a negative effect of inhibition or suppression of a metabolic activity. Induction or promotion of anthocyanin synthesis or induction of germination in seeds is a positive effect, but inhibition of hypocotyl growth or inhibition of flowering is a negative effect, both brought about by the same phytochrome exposed to red light. As we have seen till now, phytochrome plays a major role in regulating several developmental processes in plant throughout the life of the plant. In addition to this, phytochrome helps the plants to adapt to varying environmental conditions. One of the ecological functions of the phytochrome is shade avoidance. There are two types of plants, the shade plants which normally grow in a shaded environment, and the sun plants which normally grow in open fields where a lot of sunlight is available. In nature, the shade plants find the shade under the canopy of big trees. If any sun plant is shifted to grow under shade, they grow taller by showing higher rate of stem extension. This response is called the shade avoidance response. The red and infrared composition of the sunlight will be different in the open field and under the canopy. This can be measured by measuring the photon fluence rate of red and the infrared and taking the ratio of red to infrared. This measurement will show that under the canopy of other plants there will be more infrared light than red light compared with the ratio in the open field. This is because the green leaves of the trees with high content of chlorophyll absorb red light, but are relatively transparent to infrared light. The greater amount of infrared light converts the PFR to PR, reducing the level of PFR, the active form of phytochrome. This reduced level of PFR causes stem extension in the sun plants. Shade plants are those which have already adapted to this high level of infrared, and if they are shifted to open field, they do not show much reduction in their height, although the red light and hence the level of PFR will be more in the open daylight. The shade avoidance response has an adaptive value for a sun plant, as it is rapidly extending its height when it is shaded by another plant to enhance the chances of getting more unfiltered sunlight. As we have seen earlier, some seeds like lettuce seeds require light for germination. Seeds are sown under the soil where light is not available, and many of the seeds germinate in darkness and do not require light. In general, large seeds with adequate food reserves to sustain growth in darkness do not require light for germination. Seeds of cereals like wheat or maize have starch as stored food. Seeds of pulses have protein as stored food, and these seeds can sustain growth under the soil until the seedlings emerge out of the soil. Once the seedlings emerge out, they will start photosynthesis and synthesize food. But in small seeds of herbaceous and grassland species, the amount of stored food in the seeds is very less, and they are required to emerge out as fast as possible. These seeds will germinate only on the surface of the soil or at a depth of about 5 millimeter below the soil, where light can penetrate. If these seeds are buried below the depth of the soil to which light penetrates, they fail to germinate. These small seeds find the favorable depth of the soil for germination through the phytochrome, which can detect red light, gets transformed to the active PFR form and initiate germination. The seeds requiring light for germination also do not germinate under the canopy of big plants. Under the canopy, the infrared light will be higher because of the filtering of red light by the green leaves, and the phytochrome will detect the change in red infrared ratio and does not initiate germination. These seeds with small amount of reserved food always ensure that adequate sunlight is available to the seedling for photosynthesis before the reserve food is exhausted. Phytochrome helps them to detect availability of sunlight and initiate germination only when the seedlings can become photosynthetically self-sustaining. Phytochrome also regulates the sleep movement of leaves, also called as nyctinacity. In nyctinacity, the leaves or leaflets open during the daytime to face light and fold together during the night time. Plants like mimosa, albizia and samania show such type of movement. The changes in the angle of the leaves or leaflets are caused by the turgor changes in the cells of the pulvinus, a specialized structure at the base of the petiole. Nyctinacity is a type of circadian rhythm in plants, that is, the rhythmic changes that happen within a day. Phytochrome also regulates a few other circadian rhythms in plants. In conclusion, phytochrome is an important photoreceptor in plants which can influence several photoresponses throughout the life of the plant, starting from germination up to flowering. These responses are broadly referred to as photomorphogenesis, and they are independent of photosynthesis. Phytochrome induces either fast or slow responses by regulating rapid ion fluxes or by regulating the expression of genes respectively. Phytochrome also helps the plants in ecological adaptation.